This is Ed Chapman, and we're going to wrap up our discussion of the plasma membrane and the way that the fluid mosaic model works to explain the structure and function of the cell membrane. And in the first video cast I did, we talked about what diffusion and osmosis were. And in this one, we're going to focus on passive and active transport and bulk transport. And just as a quick reminder, if you think of a membrane that's semi-permeable, which means some things can cross it in either direction, diffusion primarily is the movement of small molecules like carbon dioxide and oxygen into and out of the cell. So that's diffusion. Osmosis is talking about the movement of water through the cell membrane, and this is primarily accomplished with aquaporins, which we talked about earlier. Now we're going to focus on other things like food molecules and ions, and to do that we have to talk about passive and active transport and bulk transport. Okay, before we can do this, we have to make sure you understand what an embedded protein is and why it has the shape that it has. All proteins are three-dimensional molecules, which means that they give them a certain way of interacting with things around them. And it's this three-dimensional shape that allows these embedded proteins stuck in the phospholipid bilayer to function as channels that can be controlled through the bilayer. Okay, this 3D shape allows evolution to produce different proteins that are specific to certain molecules or certain ions. And this is the idea of specificity. Different cells need to allow different things into and out of themselves at different rates and at different times. And to accomplish this functionality, they have evolved specificity, which means if you want to move ion A, then you put a lot of protein A's in the phospholipid bilayer. So the 3D shape is directly connected to their function and to this idea of specificity. And finally, these channels, these protein channels, can be open or closed. They can be activated or deactivated. So let's look at what we're talking about here. Okay, these three functions combine to produce something called semi-permeability, which is a word you've heard in connection to cell membranes probably since middle school. All right, now the first thing we're going to talk about is passive transport. And by passive, whenever you hear about things being passive in biology, that means they don't require any energy. So no energy is required. They're free. All right, and what that means is you don't need any ATP. Okay, remember, most cellular processes are driven by the conversion of ATP to ADP. And in the process of this conversion, you release a little bit of energy. And this is the power source for most of the things that happen inside of a cell. And for passive transport, you don't need this. It's free. Okay? So let's watch this video and we'll take it from there.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay, let's take that back and look at what's important here. I picked this example because it is explaining the way that synapses function. And synapses are the spaces between nerve endings and whatever it is the nerve is interacting with. So this is the nerve up here. And this could, for example, could be a muscle cell. Okay, so what this video is about is the functioning of this gap between the controlling nerve and the muscle that's going to be responding. But that's not what we're interested in right now. We're interested in passive transport. And what I'm hoping that you got to see is there are these embedded proteins in the cell membrane of the nerve that if they open, they allow a molecule to diffuse inward. So if I take the slider, let me get out of this. If I take the slider forward, you can see that once they open, calcium ions are able to diffuse inward. Okay, so that's the passive transport that we want to see. And of course, once these calcium ions diffuse inward, they interact with some vesicles in here, which causes them to exocytose their neurotransmitters out into the synaptic gap, which causes these things to open, which allows something else to diffuse inward. So we've got a kind of a chain reaction here of passive transport. And because it doesn't require any energy, it can happen very quickly. And because it's such a tiny little distance here, there's not much space between the end of the neuron here and, for example, the muscle tissue. Diffusion can happen pretty quickly because, remember, diffusion is a function of distance. So that's a good example of passive transport that touches on a lot of ideas we're going to be exploring a little bit later on. So let's get back to our PowerPoint here. All right, so that was passive transport. Let's move on and look at active transport. And active transport, remember, because the word active is invoked here, it means you do need energy. Okay, there is an ATP cost to this function. And what I want you to do is watch this video and see if you can see ATP coming into the picture. And play.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
All right, let's back this up and take a look at what's happening here. Here we have an embedded protein, here colored purple, that has a certain shape, and it's open to the inside. All right, and it's going to be functioning as a pump to move sodium ions from inside the cell outward. Now, there already are more sodium ions outside the cell than inside, so they're not going to naturally diffuse. They have to be forced. They have to be pumped. And if we take the slider and move it forward, you can see that 3 sodium ions are effectively being moved outward by the use of an ATP molecule. And the protein changes its shape. Now, once it's open to the outside, it can now pump potassium ions in the opposite direction. Now notice there already are a lot more potassium ions on the inside, so they're not naturally going to diffuse in this direction. They have to be forced. So with this open this way, they can bond to these active sites, and once that happens, you'll notice that the protein changes its shape again, and now the potassium ions can only move in the inward direction. So the net effect of all this is we are pumping sodium ions to the outside and potassium ions to the inside. But the ratio isn't perfect. It's not balanced. For every 3 going out, we only have 2 going in. So can you guess where the positive charges are going to be accumulating? On the outside. So we are separating charge. We are producing a net positive charge on the outside of this membrane, and we're leaving behind the negative charge on this side. So not only are we accumulating sodium on the outside and potassium on the inside, where we're building up a charge differential, and that's potential energy, and that's going to be important when we move forward in this course. All right, so let's get out of this, go back to our PowerPoint. All right, so active transport requires energy. Great example of this is the sodium-potassium pump. All right, now let's talk about proton pumping and co-transport. You can also pump other things out of the cell. For example, here we have an embedded protein that's using ATP energy to move hydrogen ions outward, again, putting a net positive accumulation of charge on the outside of the cell membrane, on the side facing the environment, and a net negative charge on the inside. This is building up an electrochemical gradient. Okay, so we can say that this is electrogenic. All right, in a way, it's turning the cell into like a little battery. We're separating a charge here. And this is building up something called a membrane potential. We've got a potential of voltage across the membrane. And that's going to be really important for understanding how nerves and muscles function later on. Okay, there's also something called cotransport, where in addition to pumping hydrogen ions outward, okay, and accumulating hydrogens outside now that desperately want to get inside, you can use the force of the diffusion of hydrogen ions back through another membrane protein to force or to carry or to assist the movement of other bulkier molecules like sucrose in an inward direction. So this is called cotransport because the diffusion of hydrogen ions down their gradient is being used to power the movement of another molecule, for example sucrose, into the cell. That's called cotransport. We'll stop there, and our last video cast will cover bulk transport.